For the longest time, the way we studied biology was pretty straightforward. Take things apart to see how they work. But what if the real secrets of life aren't hiding in the individual parts, but in how they're all connected? Today, we're diving into systems biology, a whole new way of looking at life as a complete functioning machine.
Okay, so for decades now, we've gotten unbelievably good at reading the genome, the complete DNA blueprint for an organism. But here's the thing, a blueprint isn't the same as the factory. The next great challenge is figuring out how to map the reactome, the entire dynamic, living network of reactions that makes a cell actually do stuff. It's the difference between having the architectural plans and walking onto the factory floor to see the assembly line running in real time.
So you might be asking, why this big shift? For so long, we just took things apart. But as we're about to see, that approach, well, it has its limits.
Right, and this really gets to the heart of it. I mean, think about it. You could study a single screw from an engine forever. You could know its exact metal composition, its thread count, its breaking point, but you would have zero clue how the engine actually generates power. Life is exactly the same. The magic isn't just in the individual parts. It's in the incredibly complex dance they do together.
And that brings us to this whole new way of thinking, systems biology. It's all about taking a giant step back so you can see the entire forest, not just one or two individual trees. So this is the big idea. It's not about just making a list of all the parts in a cell. It's about understanding that when all these parts start interacting, brand new things start to happen. These emergent properties, behaviors that you would never, ever predict just by looking at each piece by itself. So what are these emergent properties anyway? Well, think of a traffic jam. A traffic jam is a real thing, right? But you can't find it by looking at a single car. The jam emerges from the way all the cars interact. In biology, something as complex as consciousness emerges from the interactions of billions of neurons. So systems biology uses powerful mathematical models, which are fed with real experimental data, to try and understand these complex interactions and dynamics.
Okay, so how on earth do you even start to map something this incredibly complex? I mean, you can't just stick a cell under a microscope and see the whole system at once. You have to build a model.
And that, my friends, is where the computers come in. The scale here is just massive. We're talking thousands, even tens of thousands of components and interactions. The only way to get your head around that is with computational modeling. It's absolutely essential. Let's look at one of the coolest ways to do this, agent-based models. It's pretty intuitive. You basically program individual agents, like these little virtual sheep and wolves, with a few simple rules, like sheep eat grass and make more sheep, wolves hunt sheep. And from just those basic rules, you see these amazing system-wide patterns emerge, all on their own, like the classic boom and bust cycle of predator and prey populations.
All right, then you've got something called Boolean models. These are a little more abstract, but super powerful. They basically treat every part like a gene, as a simple switch that's either on or off. It sounds simple, but it's a brilliant way to map out the complicated logic circuits inside a cell that decide when a gene should be turned on or off.
And then, on the complete opposite end of the spectrum, you have these incredibly detailed kinetic models. These don't just care about on or off. No, they get into the nitty-gritty, calculating the precise rates of chemical reactions based on how much of each molecule is around. This is how you get a real-time, dynamic picture of what's happening inside the cell.
So all of these modeling tools are amazing in their own right, but there's one approach that has really taken off for getting that bird's eye view of the cellular factory, and that's mapping its metabolism.
And that brings us to this incredibly powerful tool called the Genome-Scale Metabolic Model, or you'll hear it called a GSMM.
And look at that, we're right back at our factory. This is where it all comes together. This is the actual process of drawing the complete circuit diagram, the full plumbing and wiring schematic of the cell. This is how we assemble the reactome. So what in the world is a GSMM? Well, imagine you had a giant checklist of every single chemical reaction an organism can possibly do, every way it turns food into energy or uses building blocks to make new cell parts. A GSMM is basically a computer model of that entire list. And the crucial part is, it's all built by reading the instructions written in the organism's own genome. And let me tell you, this field has absolutely exploded. The very first model, way back in 1999, was this massive achievement for just one little bacterium. Fast forward just a couple of decades, and now we have automated platforms like ModelSeed that can whip up a draft model for a new bacteria almost automatically. And today, the most advanced human model has over 13,000 reactions.
Just think about that scale for a second. It's wild.
Okay, but how do they actually do it? How do scientists go from a string of A's, T's, C's, and G's to this functional map of a factory?
Well, it's not magic. It's a really rigorous step-by-step process. You start with the genome sequence, use software to build a first draft, and then the real detective work begins. You have to find missing pieces in a process called gap filling. You have to manually check everything against what we already know from lab experiments, and then you're constantly testing it, running simulations to make sure the model actually behaves like a real living cell.
And this slide right here really brings our main analogy home. We're all kind of familiar with genome assembly, right? That's where you take all these little snippets of DNA and stitch them together to get the full blueprint. Well, reactome assembly is the next level. You're taking individual chemical reactions and stitching them together to build the complete interconnected network of the whole factory.
You see, at its heart, it's all about building from the ground up. Scientists identify one reaction, then another, and they link them all together where they share common molecules. Piece by painstaking piece, they're building this massive interconnected knowledge base of everything the cell can do.
So think about where we are. We've gone from just having the blueprint to having a fully interactive map of the entire factory. This just opens up a world of possibilities. We're talking about designing new drugs, engineering microbes to create biofuels, or truly understanding the metabolic quirks behind diseases like cancer. The question isn't, can we map the machinery of life anymore? We can. The real question is, now that we can, what are we going to build with it?